This is clip 1 of the Hallmarks of Cancer session. In this clip we will discuss the first two hallmarks, which are related to growth of cancer cells. In total, there are 4 hallmarks of cancer that can be directly linked to growth and survival of the cancer cells. We will now start with the hallmarks related to growth. The other 2 hallmarks, indicated here in orange, will be discussed in clip 2. For understanding growth and survival of cancer cells, it is however important to have a basic understanding of the cell cycle and its related definitions. Therefore, we will briefly recap this here. The cell cycle is responsible for cell division, meaning that one mother cell divides into 2 daughter cells, and this is done in different stages. Usually the cell cycle starts in the G1 phase, which is the preparation phase. Next, the cells enter the S phase, which stands for synthesis, and in which the DNA is actually copied. G2 is another preparation phase for the ultimate M phase, mitosis, in which the cells are dividing into 2 new cells. Between the different phases, there are so-called restriction points, which are there to check whether the cell still functions properly. If everything is okay, the cell cycle can continue. I will briefly introduce 3 important definitions used in this class, and these are proliferation, differentiation, and replication. Proliferation refers to growth, and it can be either physical growth, meaning that cells become larger, or it can refer to an increasing number of cells, meaning that the cell population becomes larger. Differentiation means specialization, and an example is a stem cell that becomes a specialized cell with a particular function in the liver. Finally, replication in this context means copying of the DNA, which occurs in the S phase of the cell cycle. The hallmarks that we are going to discuss in this clip are the core hallmarks called sustaining proliferative signaling and evading growth suppression. Let's start with proliferation. Sustaining proliferative signaling can also be translated as chronic proliferation. And for chronic proliferation, growth factors play a very important role. So let's have a look at the basic concept of growth factors. A growth factor can bind to a growth factor receptor, which is usually present in the cell membrane. After binding of the growth factor to the receptor, specific processes can take place, for example, phosphorylation. And phosphorylation means that a phosphate group is added to the receptor, and this results in a conformational change, a change in the structure of the receptor. For example, 2 receptors come together, form a dimer, and after that a signal is sent to the nucleus, which indicates that proliferation should be started. So growth factors are very important for this hallmark. What do we see in cancer cells? Well, cancer cells actually share a few tips and tricks in which they use growth factors to promote proliferation. And these are listed over here. So what a cancer cell can do is that it increases the synthesis or production of growth factors. Another option is that the tumor cell in itself is not increasing the production of growth factors, but the cancer cell can tell the neighboring cells to increase production of growth factors. And in this context, you have to think about the tumor microenvironment that we have discussed before. It is also possible that not the production of growth factors, but the production of the receptors at the cell surface increase. And this can also lead to an increase in proliferation. Besides that, it is also possible that the cancer cell induces changes in the structure of the receptor, meaning that there is an increased response, and that is, for example, seen in a structural change in which the receptor is always active. Lastly, it is also possible that the growth factors or the receptors are not affected, but that the cell acquires some capabilities to activate the downstream process, which is always active, and thus induces proliferation, and that is again sustaining proliferative signaling. One example that I would like to discuss, and which is very important in the field of cancer biology, is the epidermal growth factor and its receptor. You may I've heard about the epidermal growth factor and its receptor before, because a mutation in the receptor is quite commonly seen in patients with breast cancer, head and neck cancer, and lung cancer. This is also known as HER1 or HER2 mutation. So what happens in these patients is that there is a mutation in the receptor, meaning that the receptor is always active, so sending chronic signals to the nucleus that proliferation should be stimulated. So again, what we see over here, we have the growth factor and we have the growth factor receptor. When the epidermal growth factor binds to its receptor, phosphorylation can take place, a dimer is formed, and as soon as that happens, there is a downstream signaling to the nucleus indicating that proliferation should be promoted. As this receptor is sometimes mutated in patients with cancer, this is also considered a target for treatment. So as a summary, the hallmark of sustaining proliferative signaling can be seen as the gas pedal of a car that is always pushed, resulting in chronic proliferation. Let's continue with the second hallmark, which is evading growth suppression. Evading growth suppression means, in other words, ignoring the stop signal. Normally, the cell growth is tightly regulated and stopped when needed. Cancer cells can, however, ignore these stop signals. The genes and proteins that are responsible for these stop signals are the tumor suppressor genes. If the function of these genes and proteins is lost, proliferation can go on. Tumor suppressor genes will also be discussed in the next clip. If we go back to our example of the car, there's not only a problem with the gas pedal, always pushed, but there also the brake of the car is broken. One example of such a tumor suppressor gene with relevance to cancer is the retinoblastoma-associated protein. This protein is called retinoblastoma-associated protein because it was first described in children with a rare form of eye cancer, which is called retinoblastoma. The function of this RB protein is that it is a gatekeeper in the cell cycle. So let's go back to the cell cycle. An active RB protein holds the cancer cells or the cells in the G1 phase and prevents them from entering the S phase. This stop in the cell cycle is useful if there is damage in the DNA that should be repaired. If everything is okay, the RB protein becomes inactive, again through phosphorylation, so addition of a phosphate group, and the cell cycle continues. Cancer cells can have a mutation in this RB protein and the resulting protein is always inactive. The cell cycle can always continue, there is no stop signal. So what we have seen here is the relevance of growth and stop signals. Cancer cells on the one hand can have a jammed gas pedal leading to chronic proliferation. And in addition, the brake of the system is not working properly, resulting in ignorance of growth suppression.